On behalf of the Venkateswara College, I welcome you all to the fourth week session of Faculty and Development Program organized by the Venkateswara College of Pharmacy, Srikakulam, Etcherla. In previous days, wonderful sessions by Dr. A. Vasudeva Rao, International Medical University, Malaysia, and by Dr. and Koteswara Rao, by California Health Science University, and as well as by Dr. Siva S by Augusta University, and our today's speaker, Dr. B. Addagulla, Manager, Project Management of Product Development, Pharmaceuticals, Denver, Illinois, USA. Give a talk on extractables and teachables. I welcome ma'am. Now I request our principal sir to visit over the session.
Thank you, madam. Respected management of Pharmaceutical and Society, Dr. B. Sriramurthy sir, Dr. I. Shankar sir, Dr. DVS Murthy sir, and I. Kishore sir, Director of ISVC, coordinators, faculty, participants, and today's speaker, Dr. A. Swapna. Very good morning to all of you. Very good morning to all of you. Of course, Swapna madam, today it is good evening to you. On behalf of all the management members, participants, myself, a warm welcome to you, madam, to this international webinar. And 3 days, from 17th onward, we are conducting this webinar. On first day, Dr. A. Vasudeva Rao. Second day, Dr. N. Koteswara Rao. Third day, Dr. Siva Panda. Dr. A. Vasudeva Rao from Malaysia, Koteswara from USA, and Siva Panda from USA gave webinars on very good topics. And today, I request you to deliver a valuable and informative speech, that is extractables and leakables. It is very, very important topic because while formulation of a drug, after formulation of the drug into a suitable dosage form, it is to be suitably packed. So the packing is very, very important, especially in the case of the liquid formulations. It is still further important because there are many chances for extraction or leakage of the components present in the formulations. So it is a very good topic, madam. You have selected Vasudeva Rao from Malaysia. Thank you very much for accepting our invitation to participate in this event as a resource person. Thank you, madam. Over to Padma Sri, madam.
Thank you, sir. Now I request Kishore sir to address this today's session.
A very good morning to all the participants, and a good evening to Dr. Swapna. From last 3 days, I think we had wonderful sessions, and from the experts, we have heard a lot from Dr. Vasudeva Rao, Dr. Koteswara Rao, and Dr. Shiva. And today we are very much eager to hear from Dr. Swapna on extractables and leachables. So I'm pretty sure that all the participants are equally excited as me to hear from Dr. Swapna, and I hope the session will also be very useful and you would take the most out of it. Thank you.
Thank you, sir. I request P. Ramadevi Madam to introduce today's speaker. P. Ramadevi Madam. Please unmute your mic, ma'am. Please introduce today's speaker, ma'am.
Okay.
Yeah.
Audible my voice? Hello.
Yes, ma'am, it is audible. Yes, ma'am, it is
Okay.
audible. You can start.
Good morning to one and all. It's my great privilege to introduce such an eminent young and dynamic lady, Dr. Swapna Addagulla garu, innovative thinker in pharmaceutical industry with regulatory CMC. Warm welcome to you, ma'am, for SVCP webinar. Ma'am completed B.Pharm from Nagarjuna University, finished her master's in analytical from Governor State University. Nice, ma'am.
Yes.
Ma'am finished PhD in analytical chemistry, University of Rhode Island, MBA from Northern Illinois University. Ma'am has played various positions in her careers as a senior product development, analytical scientist, CMC, and regulatory liaison product development, Ocon Pharmaceuticals from August 2016 to 17. Analytical scientist, 2016 from Ocon Pharmaceuticals, Islands. Is 2014, Ocon Research Associate, Baxter International Lake, Illinois. Chemistry, Abbott Laboratories, Abbott, Illinois, 2010. A- As a part-time professor in analytical chemistry, Governor State University, Park, Illinois. And also research technician from Abbott Laboratories, Abbott, Illinois. Ma'am has polymer chemistry expertise, synthesize high molecular polymers and polymers by radical polymerization technique, and characterize products by size exclusion chromatography, viscometry, DSC, FTIR, NMR, UV and fluorescence spectroscopy. She has a vast knowledge of synthesized various silica composites, polymer silica composites, nanopolymers, composites. Ma'am has so many professional affiliations. American Chemical Society, American Association of Pharmaceutical Sciences, and Regulatory Affairs Professional Society, Pharmaceutical Management Institute. Other professional highlights are reviewer for Royal Society of History, reviewer of Journal of Physical Education, reviewer of Indian Journal of Science and Technology, reviewer of Indian Journal of Chemical Sciences. Ma'am has certifications like Regulatory Affairs Certificate, RAC in US Pharmaceuticals and Medical Devices. Thanking you, ma'am, for giving me opportunity. I'm again welcome to you, Extractable and Leachables webinar. I'm Vastu Padmasri, ma'am. Thank you.
Thank you, Ramadevi Madam, for such a nice introduction about our today's speaker, Dr. Swapna. And I welcome Swapna ma'am to take out this session and talk her about her topic. Welcome, ma'am.
Thank you very much. This is such a warm and introductory welcome. Thank you very much. And I wish everyone a happy good morning and a happy Friday. So as you all are here for the topic extractable and leachables, let us deep dive into this topic. So extractables and leachables, a little intro. So this is not a recent topic. this issue has been addressed from decades. But however, in recent years, it has gained importance because of its impact on the safety and efficacy of the drug product given to the patients or consumers. Oh, sorry. So throughout this webinar, we will be discussing about some of the outlines which are very important to address extractables and leachables. And we would be talking what are extractables and leachables, defining the terms and how it would be useful, and why should I or you or we all should be care. Why is extractables and leachables is in the news nowadays? Why is it making such a stir in the pharmaceutical industry? And we will also talk about the typical E&L processes and the various regulatory bodies of the entire regions of the world, how they have come together in unison and trying to address this topic. And in fact, the last but not least, the extractable and leachable sources. However, in this entire webinar, I will be asking you some thought-provoking questions, if you do not mind. So, how many of you know or heard the terms extractables and leachables? If you know these terms before attending this webinar, that's a good sign and it is really appreciated. And I would like yourself to give a star. So, we will be having so many thought-provoking questions down the lane. But if you know these terms and you have an idea about extractables and leachables, that is a good starting point for us. So, okay, what are extractables? Extractables are nothing but chemical compounds that actually migrate from packaging or the device materials under forced conditions. So, forced conditions are nothing but you have to use some strong solvents and a high temperature. And leachables are chemical compounds that migrate into the drug product during normal storage conditions. If you look at the pictures at the bottom of the slide, it is very self-explanatory. Extractables is nothing but the testing of the material components that comes in contact with the drug product. And leachables is we are testing the inside of the product because it has been in constant touch with the container closure system components. So where do these extractables come from? And this picture is telling us some information. If you look at those tablets are actually packed in a blister pack, and there is an aluminum foil at the bottom of those tablets packaging. And these can contribute some compounds. And the tablets are also packed in some amber colored bottles, and they have a plastic caps. Those plastic caps are made with several polymers, etc. Not only this, there are some nasal drug products which are made with the plastic container closure systems, and there are some medical devices which are inserted into the body, and those are also can leach extractables. So where do these leachables come from? We know that the drug product is in constant touch with the container closure system. But do you think just the container closure system is contributing the leachable compounds and affecting the drug product nature? No, that is not right. Because there are several other factors that actually causes these leachables. For example, on the right side of this slide, I have labeled in a blue labeled material. We always use a product that comes with a label. And there are some text and the fonts on those labels which are made with some ink. And there is an adhesive and glue that affixes these labels to the container closure system. And the studies and the research studies and the real-time studies have indicated that all these are the possible sources of the contaminants that are contaminating our drug product. Not only these CCS systems, but there are also several processing equipments. In the manufacturing of this drug product, our drug product is exposed to the filters, tubings, liners, etc. So all those are also the possible sources. And last but not least, we usually take the saline, but the saline comes in an IV bag, and there is a tubing associated with it which comes in contact with the patient. All these are the possible sources of extractables and also leachables. So, diving into the theory of extractables and leachables, we know what are extractables now and what are leachables. So, can we consider that all extractables are leachables? No. Why does a container will release all of its chemical compounds at once? But some of the chemical compounds can be released from the packaging systems and it affects the drug product. So there is always a pack and a product interaction happening on. And there are several guidelines like FDA, EMEA guidelines, which are making some significant strides to do some rational approaches of how to control these extractables and leachables. But however, I would like to raise a hand and tell that we have to mind some gap. Suppose if you have a benzyl alcohol in your container closure system, do you think that the benzyl alcohol will leach into the drug product and it will still behave as it is a benzyl alcohol? No. Some of the components, once they leach into the drug product, they can change its nature. So benzyl alcohol has the ability to form a benzaldehyde when it is in contact with the drug product. So it can change its forms from A to B or X to Y. So, why is the pharmaceutical industry is making such a big news? There is really a good reason behind it. Because this is one of the examples which I am showing in the slide. This is a very big example for the entire pharmaceutical industry. As everyone is aware, Kellogg's cereal, it is like a morning breakfast item and they come in a different type of types like a popcorns, dry cereals, etc. Whatever it is. The Kellogg manufactures these cereals, and in the year 2010, they have issued a voluntary nationwide recall of 28 million boxes of their cereals. And can you imagine these cereals were consumed by geriatrics, pediatrics, everyone. And what was the problem with that is there is some kind of some uncharacteristic odd flavor or smell that is coming from the packaging. So the patients who have consumed these cereals have been affected with nausea, vomiting, and diarrhea. And it took almost like 8 months for the Kellogg's company to determine what is actually causing this obnoxious smell. And they found out that a chemical contaminant called 2-methyl naphthalene is coming from the inner side of the packaging and is affecting the drug product. And this is one of the other unfascinating story where Tylenol, we all know that it is a non-steroidal anti-inflammatory drug to treat the pain, arthritis, etc. The same thing, it actually comes in a bottle, but bottle nobody have assumed that it would be a problematic in down the lane. The tablets are giving some odd smell, same like for the Kellogg's, and the patients are going through the symptoms of nausea, pain, vomiting, and diarrhea. This is such a truthful but bitter outcome. The source of the odor appears to be from a product of a chemical called 2,4,6-tribromoanisole. And you wouldn't believe this 2,4,6-tribromoanisole is actually a chemical that is used as a flame retardant and also used as a pesticide. So when the company manufactures the drug product in a warehouse, they put them on a pellets. And this flame retardant having 2,4,6-tribromoanisole is painted on those wooden pallets. So from the wooden pallets, it made its entry into the drug product, crossing through the secondary packaging system, the primary packaging system, and the vials and the stoppers and the labels, and it finally contaminated the drug product. So this is just for your explanation. The wooden pallets are placed on the floor of the warehouse. On top of this, all the drug products are stored. Can anyone imagine that small chemical compound from the wooden pallets gets into the drug product. And it has caused a severe financial losses to Johnson & Johnson, who was actually selling these Tylenol caplets. Those two are the most worst examples, but it is also a learning history for us not to repeat them and also to be making a judicious choice. And these are some other contaminants that we have to be aware, and it has been discovered and a control has been placed. Canadian authorities have banned polycarbonates in infant bottles, and the Germans have discovered 4-methyl-benzophenone in the chocolate muesli, which is a snack item, but still that is also being contaminated. Popcorn also. The popcorn was leached from its bag, a chemical compound called polyfluoroalkyl phosphoric acid. Not only this, to name a few, there are several other popularly used drugs like Benadryl and Motrin have also been recalled from the market for the same reasons, that there is a chemical contaminant that is affecting the safety and efficacy of the drug product. So here we are with the second thought-provoking question number 2. We all belong to the pharmacy world. So being in pharmacy world, how many of you follow the government or ISO news on recalled products and stop using them? If we are really doing this, we should give ourselves a star. And if we are not doing this, I would generally recommend all the students of the pharmacy to actually follow the news and make sure that you are updated on all the recalled products. It is helpful, and if your knowledge transfer that information to others, it helps the safety of the other consumers too. So who actually brings up all these guidelines? Who actually tells us like how to address this issue? There are several regulatory bodies in the entire region of the world, and I have mentioned here very few who have taken this initiative approach. The first one is USP, United States Pharmacopoeia, and Food Drug Administration guidelines are in place. And ICH and EP, International Content of Organization, and European Pharmacopoeia plays an important role. International Standard of Organization has recently come up with some guidelines to address this issue associated with medical devices. And several other agencies like EMEA and CHMP have a specific guidances to address the issue associated with oral inhalation nasal drug products and parenteral ophthalmic drug products. The last but not the least is the PQRI. The PQRI is like, I have to say, it is very famous and popular because this is one of the institute who has come up with several solutions for the various dosage forms. And also I'm here a little hesitant to say that though we have all these bodies working for us to address this issue, but there is no specific guidance till now that tells us exactly what to do or how to do it. Yes, this is the reality as of today, that there is no specific guidance which can guide us. So we know that these are some of the chapters that USP has come up on how to deal with extractable and leachables. They give us some basic information which is really good to know. If you look at the central triangle, which is USP 661, subchapter 1, it tells what materials are okay, because materials are made of different compounds like natural, synthetic, or semi-synthetic. So we have to make sure that if it is a synthetic material and that polymer is safe to be used on a body or not. If you look on the right side of this slide, there are some chapters called USP 1663 to 1665. Okay, so my container closure is releasing a compound called so-and-so X extractable, and it is getting into my drug product, so it is ending up as an X to Y. But is that Y really need to monitor? Yes, we need to monitor that compound because it can be causing some toxicity to the patient. So we have to do both 3 things: extractable study, leachable study, and a toxicity assessment too. On the left side of this slide, there are some chapters like USP 87 and 88. Okay, most of these drugs are initially absorbed by the human body. They go through the process of absorption, distribution, metabolism, and excretion. So there is definitely a need to assess that chemical contaminant, how it is affecting the in vitro and the in vivo studies. And last but not least here, the important thing is for a product design to get one molecule into a market, around 10 to 15 years of struggle is there because research is there, then the product development, then product manufacturing, product packaging, agency approval, and some of the molecules goes through rigorous phase studies like phase 1, phase 2, phase 3. So after spending 10 to 15 years on one product, and if you ignore addressing the extractable and leachables, it would lead to not only recall but also severe financial losses. So there is definitely a need to address this up ahead before the drug product is taken by the patient. So we've been talking about our extractables and leachables, but how to determine the extractions? Okay, a controlled extraction study is a laboratory investigation into the qualitative and quantitative attire of extractable profiles from critical components of a container closure system. So you're selecting a container closure system, make a judicious choice and make the rightest choice that the vendor is very good. You have some information about their processing aids, etc. And we also have to keep in mind our finished product excipients. In case if you have an alcohol in your final drug product, can you use a plastic container? No, because the plastic container is not suitable to close or storage the alcohol. And also, we have to make a logical selection of solvents of varying polarity. If you have a tablets bottle, which is an amber colored bottle, we generally feel that, okay, it's a glass bottle, what does it leach? No, it could still leach because of that amber nature of it. So we have to make sure that we use the solvents of different polarity so we can get that compound which can be released in the future now itself. And a case by case basis depends upon the dosage form and also the risk. Experiments to be selected for obtaining meaningful correlation. I wouldn't see that there is just one way of experimenting this information. Different dosage forms exist in the market and different experiments has to be selected. Here I'll talk about some extraction techniques. In extractable and leachable studies, there are a range of extraction techniques that can be used to either produce a solution for further analytical study or directly analyze the materials. One of the most commonly used techniques are sonication. So sonicators are available in every lab, but it does play a key role here. So if you look at the picture, that the tablet bottle is placed in a beaker with some solvent and it is exposed to sonication and with a heat on it. And if there is any chemical compound associated with the bottle, it would eventually leach into the drug product. And if I perform the testing on the drug product, then I am proactive in nature. This is the second extraction technique is a reflex extraction technique. This is also the most widely and commonly used in the laboratories. Reflex extractions, as we all are aware, are of different types. You can just do it in a separatory funnel, like a liquid-liquid extraction, separating both the aqueous and the organic layers. Or can use a reflex condenser with just the water piping going through the condensation reaction. Or you can also go for a oxalate extraction, and oxalate extraction is commonly used in United States because it is a green chemistry technique where the loss of solvent is very minimal. This slide gives much more information about the various other potential extraction techniques. Besides the sonication and the reflex condensation reactions, we can also use a sealed vessel so we can apply much more pressure to it, and also microwave assisted extraction techniques, a supercritical fluid where the water is heated at a temperature that it can take the three forms of solid, liquid, and the gas. And several headspace techniques, thermal and dynamic headspace desorption techniques are in place. It all depends upon the complexity of the formulation. If you're working on a suspension injectable, then your complexity is very high. You have to go with a much more advanced potential extraction technique. So it is a judicious choice of these extraction techniques. Okay, so we know what is an extractable and where those extractables are coming from and what are the techniques to be used. And now you have the chemical compound in your hand. How are you going to analyze? What compound is it? What is that compound that was giving obnoxious smell in the Kellogg's cereals? So analytical chemistry, which is the far most advanced and improved technology which we have the list of various equipments. So some of the techniques I have listed here on the right hand side. If you look at the headspace GCMSD, the headspace gas chromatographic technique is widely used for analyzing the volatile organic compounds. So the volatile organic compounds are usually of low molecular weight, but they do cause a potential harm. If most of you might know that the Bhopal gas tragedy is because of a small molecule called methane, and it has led to several deaths. The second technique is a GCMS called gas chromatographic mass spectrometric technique. It is a direct injection. What does the GCMS do? It actually helps in detection of several semi-volatile organic compounds such as residual monomers, antioxidant, plasticizers, etc. And the third technique is liquid chromatography, ultraviolet mass spectrometry. And this enables us to determine the non-volatile organic compounds. And these compounds are usually of a high molecular weight. This enables us to determine the largest extent of oligomers used in the preparation of the stoppers, rubber stoppers, or plastic caps, etc. Do you think the compounds only belong to these three categories? No, there are several compounds like ions, anions, and cations. Even those ions are also not safe to be consumed, and there is a need to determine those ions by ion chromatography. And elements, thanks to the periodic table, which exposes us to hundreds of elements. Does all those elements are safe? No, there are several elements which are very hazardous to the human nature, and that can be identified by using inductively coupled plasma mass spectrometry. And nitrosamines. These nitrosamines are very toxic compounds, and these can be detected by the chemiluminescence detection. The overall purpose of this slide is to give you some information like there are several organic compounds of varying molecular weights and ions and elements, and there is no one technique that can address these extractable unleachables completely. So all these techniques play an important role. Getting on to the same topic as we discussed, volatile organic compounds are nothing but low molecular weight compounds, and these are highly dangerous. There is a need to address that. And semi-volatile organic compounds are of mid molecular weight, which are easily assessed by the gas chromatographic technique. And the non-volatile organic compounds, these are easily assessed by the LC, liquid chromatography, but that must be equipped with the mass spectrometric detection technique. Because non-volatile organic compounds via LC do not have any library systems. So, if you don't look for these chemical compounds by employing all these sources, you won't find it. It is simple. If you don't look for it, you will never find for it. So we have to employ various techniques such that our product is very safe to the patient. This is the summary slide of what we have been talking from the past few slides. Various techniques to determine the volatile, semi-volatile, non-volatile, and elemental impurities. So the research does not end over here. Okay, I just do the headspace GCMS technique and it is done. No, even within the headspace GCMS technique, what should be the detection technique of my detector? Is it an electron ionization or a chemical ionization or a pressure ionization? Those also play an important role. So it is a much more deep dive topic as we go more and more in depth. Okay, once you determine the technique and also find out that yes, okay, 2,4,6-tribromoanisole has been detected. But how much amount of that compound is okay to be accepted by the human patient? So for this, thanks to PQRI, who have come up with two formulas which are very simple and very easy to understand. The safety concern threshold, also addressed as SCT, it is nothing but it is the total daily intake threshold below which a negligible safety concerns from carcinogenic and non-carcinogenic toxic effects. And the second most important formula is analytical evaluation threshold. Concentration above which the extractable or leachables needs to be identified and or quantified. So you have performed the studies and you found out that, okay, I have a benzyl alcohol, and if it is below the safety concern threshold level, we need not worry about it. But at what point I need to identify it, at what point I have to give a name to it, only when it is above analytical evaluation threshold. So these are some of the guidelines and the formulas that has been devised and which are widely used all over the entire regions of the world for oral inhalation nasal drug products. The second column, the safety concern threshold is 0.15 micrograms per day. This 0.15 micrograms per day is multiplied with the maximum daily dose of that drug product, which is already listed in the prescription. And we have to determine that if it is either below, then it is good to go with the container closure system and the drug is safe. But if it is above, then it has a separate procedure, which we discuss down the lane. Injectables. So liquid injectables, when injected into the body, it does take them some time to assimilate and give the desired action at the desired site. Hence, the safety concern threshold is higher for liquid injectables. But if you look at the last of the foremost column, which is ophthalmic solutions and suspensions, no such safety concern thresholds have been developed. So we have to do a case by case basis. So it all depends upon how complex is our formulation and what is its distribution mechanism in the body. And if it is going to reside in the human body for a shorter period of a time, then its toxicity levels are higher. But if it stays in the body and gets excreted after 5 days or 7 days, then a human can take a much more amount of any chemical compound. This is one of the example for AET calculation, and I have taken the example of a nasal spray drug product. So these nasal sprays usually come up with the plastic bottle with a tip and the tip cap associated with it, and we usually spray them into our nostrils for the desired effect. And as soon as you see the label of that container, it says that it is good for 120 labeled activations. And the dose as prescribed by the physician is a 4 activations per day. And the total fill volume of the container is 10 ml. So this formula is very clear and explanatory. Here the AET, the analytical evaluation threshold of any chemical compound, is calculated by multiplying the safety concern threshold value, which is 0.15 microgram per day, times the total use of that container, which is for 120 labeled actuations. And this divided by the total fill volume container, it is 0.45 microgram per ml, is nothing but ppm. So any chemical compound that is coming out from this nasal spray container, if it is above 0.45 ppm, then it is a threat to the patient. And there is a need to identify that compound. I had put the entire life history of these extractable and leachables and what a pharmaceutical industry would do to address this issue, and which is a very hot topic in the market industry. The top arrow, if you see, we will perform various extraction techniques as discussed earlier, either sonication or a reflux condensation, and extract the packaging component. And you will identify if there is any extractant coming from the container closure system. And if that threshold of each extract is above the safety concern, we have to send it for a toxicological assessment. And the toxicity studies will reveal the half-life period of that compound. And once we know that that compound is toxic in nature, there is definitely a need to develop that method. And not only develop, but we need to make sure that it is validated under the GMP conditions to ascertain the levels of that in our drug product. And it is not a one-time testing. We usually perform this testing as long as the drug product is going through a stability testing. And during stability testing of the drug product over a period of time, if that compound is going to be increasing along with the time, then that product is compromised. We cannot use that product. So this is like a round story of addressing the extractables and leachables. Basically, the entire life story of extractable leachables is on 4 key principles. The first principle is evaluating the interactions between packaging material and the pharmaceutical formulation and the resulting risks. We have to make a rational approaches on what is the best container for my formulation. The second one is perform an extractable study with the various extraction procedures and the different analytical technologies. We have to be very thankful to the advancement of the analytical chemistry because it has given us a various wide choices of equipment. And the third, if any chemical compound is above its threshold value, perform a toxicological assessment, defining the threshold levels for the extracted compound. And the last study is a leachable study. So when you know that the chemical compound is leaching, we have to detect it, identify, and also quantitate those leachables within the formulation, such that it does not exceed once it is released into the market, and it would not lead to recall or any other financial losses or health hazards to the patients. Okay. Here you go with the thought-provoking question number 3. Take some time and also give your response. Did any of you think or thought that container closure system can contaminate the drug product? If yes, give yourself a star. I would like to give my personal opinion on this. When I was a student in a pharmacy college, I have never thought that a container closure system plays an important role. All I was involved in is what is the quality by design, how to formulate a drug product, how to make sure that it is withstanding all the assays, purity, potency, etc. But container closure system also plays an equivalent role in the development of a drug. So here I would like to play a video of 4.5 minutes that actually gives us some insights on how to address these extractable and leachables on a practical end. And I have a disclaimer here. My disclaimer is the video which I am going to play now is not recorded by me. It is a general available video on YouTube and it is available for everyone to watch. But why I picked this video is it gives a good clear insight and helps everyone to understand the process of extractable and leachables.
Contaminants leaching from pharmaceutical container systems and product packaging has
Internet?
Swapna madam, please play the video. Play the video.
Hi, I'm playing the video. Can you hear the video? Okay, thank you very much.
Contaminants leaching from pharmaceutical container systems and product packaging has increased the awareness of the health risks posed by extractables and leachables. Offering bespoke extractables and leachable studies, Reading Scientific Services Limited can provide the necessary analytical data for regulatory submission. Here we will take you through a case study where an extractables and leachable study is required on a pediatric oral dosage format which incorporates plastic caps in the packaging. Consultations with our experts determine the details of the study specific to the requirements of the customer, such as the components of the packaging to be tested and the safety concern threshold. Based on the information gained from the consultation, stability and testing protocols are then designed which set out the specifics of the studies, ensuring regulatory guidelines are considered. To ensure integrity of analysis and results, the product in glass bottles with plastic caps is handled in such a way to limit any possibility of external factor interference. For example, the adhesive on the label may contain compounds that can leach into the drug product. The next stage is to begin the leachable study, which requires the sample to be stored under the conditions set out in the protocol. Our stability cabinets enable storage of samples under accelerated conditions, including temperature and humidity, which are designed to replicate what the product may encounter in its lifespan. The extractable study focuses on determining the chemical compounds present in the plastic cap that have the potential to leach into the drug product. To ensure complete extraction, the surface area of the sample is increased by cutting the cap into pieces. It is then subjected to harsh extraction conditions. These can include refluxing in different solvent combinations. We have the capability and experience to employ the most appropriate extraction techniques to recover all potential extractables from your sample. The resulting extraction solutions are then analyzed using analytical techniques which are capable of detecting a wide range of compounds. Inductively coupled plasma mass spectrometry, ICP-MS, is used to screen the sample for up to 70 different elements, including heavy metals. This technique is sensitive and robust, allowing for the simultaneous qualitative and quantitative analysis of all elements present. Following the elemental analysis, the sample is then analyzed for organic compounds using mass spectrometry. Two techniques are employed, one being gas chromatography mass spectrometry, GCMS, and the other being liquid chromatography mass spectrometry, LCMS. GCMS analysis is performed by using both headspace and direct injection, as this allows for detection of volatile and semi-volatile compounds. Compounds soluble in water are analyzed using LCMS, which has increased sensitivity over a large mass range. Once the extractables profile of the bottle cap has been determined, the leachables analysis can be performed. The leachable study will determine if any of the extractables identified have migrated into the drug product in sufficient quantities to be of concern. The samples stored in the stability cabinets are removed at the agreed time points, and analysed by ICP-MS, GCMS, and LCMS. We assess the results by applying an analytical evaluation threshold in order to ascertain if any of the leachables are at a concentration that will require toxicological evaluation. This threshold is based on the regulatory guided safety concern threshold, the analytical technique employed, and the drug product configuration. We deliver an extensive risk assessment throughout the extractables and leachables investigation to meet the regulatory requirement. By providing comprehensive extractables and leachable studies, we can give our customers the confidence that they are ready for regulatory submission.
Thank you very much. So that was the video. I hope it gave some insights in general how we perform the extractable leachable study in the laboratory. The main reason I would like to bring that video is most of the people who are very good at theoretical have problems in understanding the practical implications. So that is how the video has been depicting the studies for the extractables and leachables are done. So in the conclusion, I would like to just mention that the quality by design, the QBD, which is the foremost thing that is happening in the pharmaceutical industry, that approach can provide a framework for a more scientific risk-based approach to the extractables and leachables. And a proper container closure system will always pay off better in the longer run to avoid recalls and financial losses. There is a need to address extractable and leachables, considering the safety of the patient and consumers. And lastly, I would like to acknowledge my mentor who has trained me on extractable and leachables. He is none other than Dr. Dennis Jenkins. He is the chairperson of the Product Quality Research Institute. I would also like to thank from the bottom of my heart to Mr. Prashant Damarasinghu. He is the associate professor at the Venkateswara College of Pharmacy, and also to the principal and Dr. Kishore sir for giving me an opportunity to execute this webinar to the pharmacy college. And as you know, I've been explaining in the webinar, we have come across the 3 thought-provoking questions, and there is a star associated with it. So whoever has scored either 1, 2, or a third 3 stars, kudos to you all. Something is better than nothing. If I were in your shoes, I might have not scored a single star. So those who ever have scored a single star, you are still the heroes. So I would like to end my presentation with a philosophical quote, which I really like, and that is from Mr. Aristotle. The more you know that you don't know more. So there are several topics worldwide, and the more you deep dive, we know that we don't know more. So this is one of the topic, extractable and leachables, but there is definitely a need to address this issue. And thank you everyone who have been directly and indirectly involved in bringing this webinar in front of you all. And considering this new coronavirus, I would expect everyone to stay safe from COVID-19. And I would leave the floor for questions.
Thank you, madam. You have given a very wonderful nice and elaborative and informative topic because um, how much importance the drug designing, drug development, and product formulation is having more than that one. This analysis of the extractables and leakables is having because these extractables and leakables, if they are not within the limits in the package material or the containers, whatever it may be, that may lead to very serious effects and that will contaminate the product and that will lead to very serious effects to the patients. Thank you, madam. It is very, very interesting topic. Um, Thank you very much for giving such a nice presentation. Now I request Padmasri madam go for the question and answer session. Padmasri.
Yes, sir. Yes, sir. Thank you, Madam Swapna, Madam. Here we are having some questions from the participants. And the first question is from Satish Ponanda. He is asking that if chloroform is used as solvent in extraction, in presence of light, it converts into phosphazine, which is very toxic. So how it can be prevented?
Yes. So in general, the most commonly used solvents are of low boiling point. And we have to make sure that the low volatile organic compounds have a molecular weight less than 100. So chloroform we can still use, but it is quite evaporative. Even with this oxalate extraction, the extraction is not possible. And I do understand because of the presence of the chloroform, it ends up into a phosphazine compound, etc. So there are issues. Uh, for his purpose, I would like to say that the most commonly used solvents are methanol, hexane, IPA, dichloromethane, and the water mixed with the ethanol, 70:30 ratio. I don't know if I have answered his question. I hope if it is not clear, I can try to explain in another way.
I hope he got his doubt clear. And another question is from Nakka Rajeshwari. How extractables and leachables are related to pharmaceutical impurities? I hope at the starting session she questioned this, the starting of this session. During the session, she might have cleared her doubt, but anyway, once again, please you clarify her doubt.
Yes, yes, yes. That's actually a very good question. I have to say thanks to her. That's a very good, insightful question. So, these chemical contaminants, such as extractables and leachables, are very different from impurities. So, impurities, there are guidelines such as ICHQ3 2A for drug substance, ICHQ3B for drug product. And those impurities are quantitated according to the identification threshold and the qualification threshold. But whereas the chemical contaminants from the extractable leachables, there is no such particular guidance and we go only with the safety concern threshold value as given by the PQRI. So impurities within the drug product is a separate thing and the impurities that are coming from the container closure system are something different. These two have to be addressed and detected in a separate ways.
Okay, thank you, ma'am. And so many participants like Meghna Patnaik, Gupta, Reshma, Kurmana, and some other people also asking the same question like when should an extractable and leakable study should be performed?
Very good question. Yes, that is the right question too. So when you have developed your product formulation and you are doing your clinical studies. So your product, you have some product in the back end which is sitting in the research and development area. So now you can make a judicious choice of your container selection because these clinical phase studies does take time. By the time those studies results come in, you can now challenge your drug product with various container closure systems and put them on a stability and then determine if it is leaching any component. And in case if you have a molecule that doesn't go with the clinical studies, then you have very shorter time. In that case, I would say that you expose your container closure system components with the various solvents and do a very forced, vigorous extraction conditions, like expose it to the higher temperature, higher pressure, higher humidity, or expose it to the brightened sunlight or UV visible light, and then see what could be the worst possible future contaminant that it could leach.
Yes, ma'am. Accelerated studies like that.
Yes, exactly.
Okay, ma'am. Next question is from Anusha Narayan Shetty. What is the safety level of extractables and leakables?
Can you repeat the question again, please?
What is the safety level of extractables and leakables? Is the question.
Yes. So, in one of my slides, I mentioned that for orally inhaled and the nasal drug products, the safety concern threshold is 0.15 micrograms per day, and that is also multiplied with the maximum daily dose of our drug. But in case if it is an injectable formulation, it is 1.5 micrograms per day. And there are several other formulations like suspensions, emulsions, transdermal systems, and subcutaneous patches, etc. But for those, none of the guidances in the entire region of the world came up with some threshold values. So the constant one which we use in our regular scenarios is 1.5 micrograms per day.
Okay. And thank you, ma'am. Another question is also from Satish Kondada itself. Because of chemiluminescence, there is a chance of degradation of extraction of compounds. Then how it can be managed? Is there any
Yes.
chance of such type of degradation or not?
There is definitely there. So what happens is I have a compound like let us say Benadryl injection, and if I try to do the chemiluminescence detection, what it will happen? So the main ingredient of my drug product, it would be broken down into several pieces, and one of that piece that I was looking for is nitrosamine. But at the same time, it is also giving me several other chemical compounds. So one technique for each compound. So if you're only looking for nitrosamines, just look for that in the chemiluminescence and forget about all other possible degradation products that come in that. And if you want to just look for some low volatile, with the H-based chromatographic techniques. So that's why only not one technique is there. There are several techniques to address each type of specific compound. But he is right. If you apply more stress, it would lead to more degradation. Sometimes more degradation is third.
Another question is there from and Rajeshwari. In quantification studies of impurities, which one is a better way to apply relative responses, relative response factors, or to use impurity reference standards?
Okay. Uh, that is really a good question. But for extractable and leachables, we do not even put RRF factors. But for her reason, I do have some knowledge on it because I'm also a validation scientist in my background. It is always good to use the RRF factors. Okay, relative response factors because you actually take that reference standard and you perform the linearity from the 50 to the 150% of the assay level, and you also perform the precision and the accuracy. You do the spiking study and you get the accuracy recovery values. And from that, you are depicting the formula for RRF and you are getting one quantity for that. And I always prefer RRF in the real life world because this product we are going to manufacture for the whole lifetime. And if you don't find that reference standard in the market, then your drug product testing is at stake. So it is always good to use RRF factors instead of reference standard.
Okay, fine. Another question is from Venkateswara Demon. What is the relationship between elemental elemental purity in extractables and liquids?
I have a disturbance. Can you please repeat it again?
Yeah. Yes, ma'am. What is the relationship between the elemental entity and elemental impurities?
Oh, okay. Yes, yes. Okay. So elemental entities, suppose if you're thinking about a silver, but a silver do not exist like a silver ion as in the extractables and leachables. It usually comes in a complexation form like a silver chloride or a silver EDTA complexing chelating agent. So that is called as an elemental entity. And we cannot detect the elemental entities by any detection technique. Because we suppose if you're looking for a silver chloride, chloride can be determined by the ion chromatography technique, and the silver has to be detected by either atomic absorption spectrometry or inductively coupled plasma mass spectrometry techniques.
Okay. And I hope it's the last question, ma'am, from Bora Gupta. How does a risk assessment assist when evaluating extractables and liquids?
That is very good. A very good question, I have to say. So, how we assess? Okay, what is it? Is okay. I'm actually developing a drug called suspension, and my suspension, the physician always recommends to send that suspension in a pre-filled syringe form. Okay, so that is keeping in the mind of the physician's suggestions. I'm developing my suspension. So I'll make sure that whatever the manufacturing equipment that comes in contact with the manufacturing of a suspension, okay, filters, tubings, and okay, I'll go to the filter vendor and ask him like, how did you prepare your filter? What are the processing aids used in your filter? What are your plasticizers, antioxidants? And all this information is gathered from the vendor and put into a risk assessment and see what are the possible compounds that could come from those manufacturing operations. The same task is also executed with the raw materials and the excipient vendors and also the container closure components. So I go to the syringe, barrel, and the rubber stopper that is present in the paper syringe vendors, and I get the possible information from them. And we actually tabulate all the possible components from the various vendors' components, and we will make sure that okay, one of the guy has used some kind of a trinitrotoluene as an aid in the development of a stopper. And I'll keep my eye that, okay, when I use my container with the drug product, I put them into headspace, sorry, not direct GC chromatographic technique, and we'll try to look for that trinitrotoluene. Oh, is it detected? It's not detected. That means my drug product is free from such contaminants. It is a much more in-depth, deep dive topic, but yes, there is definitely a need for that risk assessment from the various regions, from API, excipient, manufacturing components, container closure systems, and the dilution.
Okay, ma'am. There are no more questions from the participants. Thank you very much, ma'am.
Obviously, I think there one more question from Bhaskar Dunna. D. Bhaskar. I think he was asking about
Madam.
what are the major adverse.
Yes.
Okay, let me take it. Yeah. So the question is, what are the major adverse effects if a person took extractable and leachable product?
Can you repeat the question?
Yeah, he's asking what are the major adverse effects if a person took extractable and leachable product? I mean, if a patient.
Yes. There are several major adverse effects depending on the type of chemical compound that he is taking. That's why I gave you the real life examples. For Kellogg cereal, when he was taking that cereal, he's affected with a chemical called 2-methyl naphthalene, and that will induce nausea and vomiting. And whereas the Tylenol was also giving diarrhea. But so far, luckily, we haven't seen any serious deaths with these chemical compounds. So adverse effects are there, but those are still manageable, and people have been hospitalized. And human bodies are very sensitive to each and every chemical compound, so their sensitivity cannot be addressed ahead.
Okay. I hope that answers.
Hello, madam, one more question.
Yes, sir.
Uh, when the extractable and leakable test should be performed?
Yes, sir. That question is asked by so many persons, and I
Yes.
already posed it, sir.
Okay. Okay. Then it
Madam
is.
already answered to that question.
Okay. Okay. Then it is okay.
Okay, sir. Thank you, ma'am, for your valuable session. And now I invite Mr. Prashant, sir, for vote of thanks.
Padmasri. Padmasri. Padmasri, madam. Hello.
In fact, I mean, it was very detailed and I mean, the slides were very informative. Thank you so much.
Thank you very much, sir.
Good morning, sir. Good morning, sir. Good morning to all. It's our pleasure to have you all participants for this 4-day International E-Faculty Development Program with all eminent personalities organized by Venkateswara College of Pharmacy, Etcherla, in association with ISVC. I am very thankful to a young dynamic speaker, Dr. A. Vasudeva Rao Garu, for his talk on e-learning and collaborative learning. And I'm also thankful to Dr. Koteswara Garu, an eminent personality who talked on the exosomes and its activity on the cerebral ischemia. I'm also thankful to another eminent personality, Dr. Siva S. Panda Garu, for his valuable knowledgeable speech on rational approach on drug design. I am also thankful to our today's young and dynamic speaker, Dr. Swapna Addagulla, for her talk on extractables and leachables, gives exposure to the industrial level. I am also heartily thankful for our management and patron of this 4-day international webinar, Faculty Student Development Program, Dr. A. Murthy Garu, Dr. Murthy Garu, and Dr. I. Shankarao Garu for their readily accepted to conduct this FTP program in successful manner. I thank to our principal sir and convener of this FTP session, Dr. K.V. Nagoji Garu, for his involvement and support in all aspects to conduct this FTP program in a successful manner. I especially thank Dr. I. Kishore Garu, Director of ISBC and co-convener of this FTP program, for his timely support and in every step and organizing this program in a smooth and successful manner. I heartfully thank to all faculty members who supported their valuable time and also their coordination to conduct this FTP program in a successful manner. And I'm also thankful to other members who also played their role in the different aspects and help in their help in the grand success of this FTP program. Last but not least, I thank all the participants who have actively participated and made this FTP program a grand success. Thank you to all. I hope our 4-day international webinar resulted in good extraction and sharing of knowledge and emerge as the best knowledge sharing platform. Thank you all for your support and thank you all. Thank you, sir. Now, hand over to our principal sir to complete this session.
Okay, thank you, Prasad.
sir.
Thank you, Prasad. Nothing will be there after telling vote of thanks. So, you addressed all. Um, and I am very much thankful to all, each and everyone, those who have contributed for the success of this webinar. Thank you, one and all.
Thank you, sir. Thank you to hello, Dr. Asma.
Yes, sir.
Yeah, Swapna. Thank you, Swapna, for your back.
Thank you, Prashant. It was a great pleasure working with you.
Thank you, thank you.
Thank you very much.
Now I'm going to complete it. Thank you all.
Thank you, everyone.
Thank you, sir. Kishore, sir.
Thanks, Prashant. Yeah. One second. Okay, fine. I think we just went. Thank you, Dr. Swapna. It was a wonderful session. Thanks, Prashant. Yeah. One second. Okay, fine. I think we just went. Thank you, Dr. Swapna. It was a wonderful session.